Hello, everyone. My name is Scherer. I'm a postdoc at the Center for Genomic Regulation in Barcelona and was formerly at Saarland University. Today, I would like to present you here at this virtual poster presentation with my project entitled Identification of Tissue-Specific and Common Methylation Quantitative Trait Loci in Healthy Individuals using MAGAR. So let me first introduce to you what a methylation quantitative trait locus, methQTL, actually is. So you imagine you have this population of people here. They have a particular genomic constitution at this location. They have two alleles, and both of these alleles show the same genotype. Now, at some location that can be far away or more close to it, for the sake of this project, we only consider associations within 1 MB of this genetic position. There is a DNA methylation state, which is in this case fully methylated. And now you do the very same thing in another population of people that have a different genomic constitution. So one of the alleles actually shows a different base at this particular location. So this is a single nucleotide polymorphism that has different potential alleles. And here you have a heterozygous situation. And it turns out that at this very same genetic location, epigenetic location, you have a loss of DNA methylation of parts of the molecules. So there is a very strong association between the DNA methylation state and the genotype here. And this genotype that is associated with the DNA methylation state is referred to as a methQTL. There is not really one package available to identify these methQTLs from raw data. This is why we developed the R tool called MAGAR, which stands for Methylation Aware Genotype Association in R. And on the left-hand side here, you see the overview of the MAGAR package. It is in general a two-phase approach and quite complex. So let me guide you through it in a bit more detail. We first start with this raw data, which can be either microarray based or also sequencing based, and then use established tools such as RNBeats, CRLMM, or P-Link to do data import and to end up with a DNA methylation data matrix, as well as with a genotype data matrix. MAGAR also supports automated imputation using the Michigan imputation server. Then in the second phase of the tool, we actually use the methQTL calling or the identification process. And this is again another two-stage approach. First, we leverage the properties of DNA methylation data. That is that neighboring CBGs are typically very highly correlated. This is why we can group them together in so-called CBG correlation blocks. That's what we do here in stage 1 by first computing a correlation matrix, then weighting these correlations according to the genomic distance, and employ Louvain clustering to identify the CBG correlation blocks. In the second stage, we then compute the methQTLs by first determining a tag or representative CBG per correlation block, and then associate this DNA methylation state of this tag CBG with all genotypes in this given genomic distance, which is typically 1 megabase. And this is conducted using linear models, which will return you an effect size, which is the strength of the effect of the genotype on the DNA methylation state, as well as an associated p-value. Within this project, we went a step further and actually wanted to ask the question whether the methQTLs that we determine are rather tissue-specific or shared across different tissues. This is why we collected in the context of the CISCIT consortium from healthy individuals T cells as well as B cells and ileum and rectum biopsies. We then apply MAGAR to each of these tissues or cell types separately to obtain these statistics. So these methQTL statistics are the association between a SNP and the CBG, as well as the associated effect size estimate, so the strength of the association, as well as an associated p-value. We now wanted to ask the questions whether these methQTLs that we identify individually are shared or tissue-specific, and we used colocalization analysis to ask this question. More specifically, we used colocalization analysis as implemented in summary data-based Mendelian randomization. And this will basically tell you whether an association that you find individually is shared or rather tissue-specific. And since we do this in a pairwise manner across all of these tissues or cell types, we will end up with two classes of methQTLs, those that are shared across the tissues and the tissue-specific ones. When we apply this to our data set, we find that most of the QTLs are actually shared across tissues, while only some of them are tissue-specific and preferentially in T cells. We then investigated the specific properties of tissue versus shared methQTLs and found that the shared methQTLs are actually stronger. So the effect on the DNA methylation state is stronger, which we can measure using the effect size, while the distance between the CBG and the SNP is not different in the two classes of QTLs. We also asked the question whether these tissue-specific or shared QTLs are located in particular parts of the genome, and found that the tissue-specific methQTLs are preferentially located in proximal enhanced elements. So as a summary, I presented to you MAGAR, which is our software package for the identification of methQTLs from raw genotyping and DNA methylation data. We used MAGAR and colocalization analysis to determine tissue-specific and shared methQTLs, and found that tissue-specific methQTLs are actually preferentially located in proximal enhanced elements. MAGAR is available as an R package from a bioconductor. So I would be very happy to discuss this whole project with you in much more detail, so don't hesitate to visit me virtually at my poster here, or you can also check out our preprint, which is available from the bioRxiv. Thanks a lot for your attention.